endocytosis and lysosome function. One of the major functions of lysosomes is the digestion of material taken up from outside the cell by endocytosis. However, the role of lysosomes in the digestion of material taken up by endocytosis relates not only to the function of lysosomes, but also to their formation. In particular, lysosomes are formed by the fusion of transport vesicles budded from the trans-Golgi network with endosomes, which contain molecules taken up by endocytosis at the plasma membrane. The formation of lysosomes thus represents an intersection between the secretory pathway through which lysosomal proteins are processed and the endocytic pathway through which extracellular molecules are taken up at the cell surface. Material from outside the cell is taken up in clathrin coated endocytic vesicles which bud from the plasma membrane and then fuse with early endosomes. Membrane components are then recycled to the plasma membrane and the early endosomes gradually mature into late endosomes, which are the precursors to lysosomes. One of the important changes during endosome maturation is the lowering of the internal pH to about 5.5, which plays a key role in the delivery of lysosomal acid hydrolases from the trans-Golgi network. Acid hydrolases are targeted to lysosomes by mannose 6-phosphate residues, which are recognized by mannose 6-phosphate receptors in the trans-Golgi network and packaged into clathrin coated vesicles. Following removal of clathrin coat, these transport vesicles fuse with late endosomes and the acidic internal pH causes the hydrolases to dissociate from the mannose 6-phosphate receptor. The hydrolases are thus released into the lumen of the endosome while the receptors remain in the membrane and are eventually recycled to the Golgi. Late endosomes then mature into lysosomes as they acquire a full complement of acid hydrolases which digest the molecules originally taken up by endocytosis. Phagocytosis and autophagy. In addition to degrading molecules taken up by endocytosis, lysosomes digest material derived from two other routes, phagocytosis and autophagy. In phagocytosis, specialized cells such as macrophages take up and degrade large particles including bacteria, cell debris and aged cells that need to be eliminated from the body. Such large particles are taken up in phagocytic vacuoles or phagosomes, which are then fused with lysosomes, resulting in digestion of their contents. The lysosomes formed in this way can be quite large and heterogeneous, since their size and shape is determined by the content of material that is being digested. Lysosomes are also responsible for autophagy, the gradual turnover of the cell's own components. The first step of autophagy appears to be the enclosure of an organelle in membrane derived from the endoplasmic reticulum. The resulting vesicle then fuses with a lysosome and its contents are digested as described in the image. Autophagy is responsible for the gradual turnover of cytoplasmic organelles. Peroxisomes. Peroxisomes are small membrane enclosed organelles that contain enzymes involved in a variety of metabolic reactions, including several aspects of energy metabolism. Although peroxisomes are morphologically similar to lysosomes, they are assembled like mitochondria and chloroplasts from proteins that are synthesized on free ribosomes and then imported into peroxisomes as complete polypeptide chains. Although peroxisomes do not contain their own genomes, they are similar to mitochondria and chloroplasts in that they replicate by division.